Every cell in our body reads instructions from DNA. Genes tell cells what to make, how to act, and when to grow. Sometimes a tiny change appears in a gene, and this is called a mutation. So, mutations are common and happen in all living things. Some cause visible changes, some cause diseases, and some makes no difference at all. Now let's revise what DNA is. Deoxyribonucleic acid. It is the chemical that carries all the information needed to build and run our body. It is found inside the nucleus of almost every cell and help decide our traits like height, our eye color, and many other functions. It has a double helix shape which looks like a twisted ladder. It is made up of small units called nucleotides which join together to form long strands. Small sections of these strands are called genes, and genes give specific instructions to our cells. DNA is packed neatly into structures called chromosomes. They are tightly coiled bundles of DNA that are DNA is wrapped around proteins to keep everything organized. Humans usually have 46 chromosomes arranged in 23 pairs. Our genes are located on these chromosomes, carrying the instructions that tell our body how to grow, develop, and function. Genes are used to make proteins, which are the building blocks and workers of our body. Proteins are made from small units called amino acids, and they do almost everything from building tissues, helping chemical reactions, sending messages, and defending us from germs. In short, DNA is the plan, genes are the instructions, and proteins, which are made up of amino acids, are the result that keeps our body alive and working. Now, how does DNA actually make proteins? This is explained by the central dogma of molecular biology. So, DNA first make copies of its instructions in the form of RNA in a process called transcription. Then the RNA carries these instructions to the cell's protein making machinery, ribosomes, where proteins are built in a process called translation. So the information flows like this from DNA to RNA and then to protein. And this is how our cells turn genetic instructions into the molecules that keep our body alive and working. Now, sometimes a tiny change occurs in a gene or DNA sequence, and this change is called mutation. Um, it's like a small typo in the body's instruction book. Mutations can happen randomly when DNA is copied, or they can also be caused by things like radiation or certain chemicals called mutagens. Some mutations change how our cells or proteins work. Some cause diseases, and some have no effect at all. Genes can change in several ways, creating different types of mutations. So, there are point mutations where only the single nucleotide in the DNA is changed, and then there is substitution in which one nucleotide is changed by the other nucleotide. Like, for example, um, AAG is changing to ATG. So here the A changes to T. And then there are frameshift mutations in which the nucleotide is added or removed, which shifts the reading frame of the whole DNA sequence. So the amino acid which is which were made earlier are now changed, and they are the different there's the whole different series of amino acids which completely changes the protein which is being made. And then there is insertion, where an extra DNA letter or nucleotide is added to the sequence, and then there is deletion, where the DNA nucleotide is removed from the sequence. Now, these changes can affect how proteins are made and may change how the body works. So, genes tell the cells how to mm, combine, how to link amino acids to make proteins. So, if a mutation changes the DNA, it can also change the sequence of amino acid in a protein. So, there is a completely different protein which is being made after mutation. So, when the amino acid sequence is altered, the protein's shape may change, and since the protein shape is important for its job, this can make the protein work incorrectly or change its function entirely. Some mutations are harmful and can cause problems in our body. They may produce proteins that do not work properly or disrupt important processes in cells. Harmful mutations can lead to diseases such as cystic fibrosis, sickle cell anemia, or certain types of cancer. Cystic fibrosis is a disease which is caused by harmful mutation in a gene called CFTR, and this mutation changes the protein that controls the movement of salt and water in and out of the cells. So, as a result, a thick mucus builds up in the lungs and digestive system, leading to breathing and digestion problems. Now, not all mutations are harmful. Some mutations can also be beneficial and give an advantage to an organism. For example, certain mutations can make people resistant to diseases or can help bacteria survive through antibiotics or allow plants to adapt to new environments. Beneficial mutations are rare, but they play an important role in evolution and adaptation. On the other hand, many mutations have no noticeable effect on the organism, and these are called neutral mutations. So they can happen randomly when the DNA is copied, or they may be caused by environmental factors like radiations or chemicals. Sometimes, even if a mutation changes a DNA nucleotide, the protein remains the same because there are many codons which codes for the same amino acid. Even though they do not cause any harm or provide a benefit, neutral mutations can be passed on to future generations and add to the genetic diversity of a population. Over time, this diversity can help a species adapt to new environment or changes in their surroundings. Neutral mutations quietly occur in our DNA, contributing to evolution without causing any obvious changes in the organism. Using models is very helpful for understanding complex scientific ideas like gene mutations. Models can be simple, like diagrams or paper cutouts, or can be more advanced, like block constructions or computer modeling or simulations. All of them help how changes in DNA can affect proteins. So there are benefits of using models because the models help